welcome back to another GCSE Biology by SmartPath Tutoring. This video is on transpiration and stomata. Transpiration rate is affected by four main things. Number one is light intensity. The brighter the light, the greater the transpiration rate. Stomata begin to close as it gets darker. Photosynthesis can't happen in the dark, so they don't need to be open to let CO2 in. When the stomata are closed, very little water can escape. Number two is temperature. The warmer it is, the faster transpiration happens. When it's warm, the water particles have more energy to evaporate and diffuse out of the stomata. Number three is airflow. The better the airflow around a leaf, for example stronger wind, the greater the transpiration rate. If the airflow around the leaf is poor, the water vapor just surrounds the leaf and doesn't move away. This means there's a high concentration of water particles outside the leaf as well as inside it, so diffusion doesn't happen as quickly. If there's good airflow, the water vapor is swept away, maintaining a low concentration of water in the air outside the leaf. Diffusion then happens quickly, from an area of higher concentration to an area of lower concentration. Number four is humidity. The drier the air around a leaf, the faster transpiration happens. This is like what happens with the airflow. If the air is humid, there's a lot of water in it already, so there's not much of a difference between the inside and the outside of the leaf. Diffusion happens fastest if there's a really high concentration in one place and a really low concentration in the other. Measuring transpiration. Potometer experiment. You can estimate the rate of transpiration by measuring the uptake of water by a plant. This is because you can assume that water uptake by the plant is directly related to water loss by the leaves. Transpiration. Set up the apparatus as in the diagram and then record the starting position of the air bubble. Start a stopwatch and record the distance moved by the bubble per unit time, for example, per hour. Keep the conditions constant throughout the experiment, for example, the temperature and the air humidity. Potometer diagram labels. Plant cutting sealed in a tube. Reservoir of water at the top with a tap labeled "Tap is shut off during experiment." Text arrow, as the plant takes up water, the air bubble moves along the scale. Text arrow, water moves this way. Capillary tube containing an air bubble, labelled, bubble moves this way. Capillary tube with a scale. Beaker of water at the bottom end. Guard cells are adapted to open and close stomata. They have a kidney shape, which opens and closes the stomata in a leaf. When the plant has lots of water, the guard cells fill with it and go plump and turgid. This makes the stomata open so gases can be exchanged for photosynthesis. When the plant is short of water, the guard cells lose water and become flaccid, making the stomata close. This helps stop too much water vapor escaping. Thin outer walls are thickened inner walls, and thickened inner walls make the opening and closing work. There's also sensitive to light and close at night to save water without losing out on photosynthesis. You usually find more stomata on the undersides of leaves than on the top. The lower surface is shaded and cooler, so less water is lost through the stomata than if they were on the upper surface. Guard cells are therefore adapted for gas exchange and controlling water loss within a leaf. That's the end of the video. I hope you enjoyed.